First of all, I would like to thank Telefónica, as well as the Guggenheim Museum, and Professor Lehn for being here. Our education minister, Christina, talked about Bertrand Russell, quoting him, saying that science as a prosecution of the truth is nor better nor worse than art, but through technology, the power that it has to transform the economy is very difficult to be compared to. So saying this in such a wonderful art venue might seem to be a blasphemy, but it is true that the North American economy has grown in a 2% yearly in the last 130 years in average, which is an exponential growth, and studies show that it is mainly due to technology. So we have Solo, Canaro, and so on and so forth. And it was also explained that science was three things: culture, and it is an essential part of culture because we want to understand things. It is also economy. The economic growth is exponentially in a cycle that is propagated by science and technology. And it is also training. Those that have studied science have skills that transmit creativity and innovation to the production system. That's why in the Donostia International Physics Center, in the PhD, and in the Excellence Campus, we believe that contributing to a society that is scientifically informed is not only profitable, but it is our duty, because this society will be freer, more democratic, and won't be able to be manipulated by any kind of interest. That's why we're here today with an excellent speaker. This is an activity organized in Vizcaya by the Berg Center, the Donostia International Physics Center, which is a private-public partnership alliance. The university, the town council, and the regional government are part of this center, as well as Naturgas, CAF, and Kutsabank, and especially as our host today, Telefónica. Telefónica, that since the starting point, has been giving us their support, especially the director. And also, this center is not only a center of the Basque University, it is a center that it is also linked to research in the Basque Country, also through the Excellence Campus. And together with Tecnalia, Jauregui is here. And we also have the rector here, and they all foster this Excellence Campus. The director of the PhD for Scientific Culture, Iñaki Pérez, is here today, and we want to be more and more present in Vizcaya because we are a Basque center, not only a center for San Sebastian, and we feel very welcome here in the Central Rectorate, and we are very grateful to our rector because after Javier Benito's presentation, he will be introducing Marie Lehn, who is a very good friend of the Basque. He's visited us on many occasions, and he was also awarded the Nobel Prize, and he is a counselor for one of our centers. Therefore, I would like to thank you for your presence here today. I would like to thank those that have come from outside the Basque area, and I would also like to thank the speaker of this morning, and I would like to give the floor to the director of Telefónica here in the Basque Country. Good afternoon. I would like to say hello to the rector, governmental representatives, and friends. It is a special day for us today because we're able to visualize our commitment with Bilbao, Vizcaya, and the Basque Country. Telefónica and Telco Digital want to be present locally. We're at the Guggenheim Museum, and this is an event organized by the Donostia International Physics Center, and we support and are committed with these two organizations. So this allows us to show our social responsibility with both art and with science in the difficult times we're living. Telefónica has a very long-standing committee with the Basque Country. We are carrying out investments in 4G and fiber that will allow not only the development of the Basque Country at the head and as a reference for Europe, but also supporting the museum and the Donostia International Physics Center so that this knowledge is better known, and we also would like to foster the international image of the museum. But it's not the time to speak about Telefónica. It's time to listen and learn from Jean-Marie about science, and it is wonderful to listen to how he explains his knowledge and reflections. Today is a beautiful day for all. There's a very good chemistry between the Basque Country, culture, and chemistry, and as I was saying, science. So thank you. I would like to, first of all, say good afternoon to everyone.
The world's leading authorities in his field. I would like to welcome you. Thank you for having agreed to give this lecture, and particularly thank you for your long-standing close relationship with Donostia International Physics Center and with the University of the Basque Country. Professor Lehn is not a great scientist only thanks to his research, but that is also down to the dynamic and entertaining way that he shares his knowledge. That is why we are very lucky today. El profesor Jean-Marie Lehn nació en Rosen.
Professor Jean-Marie Lehn was born in Rosen in France and studied chemistry in the Strasbourg University. He worked together with Robert Pons Woodward in the Harvard University, developing chemical synthesis of the B12 vitamin. Then he got a position as a professor in Strasbourg, and in 1980 he was professor of the prestigious Collège de France in Paris. In 1997, he shared the Nobel for his studies of the chemical base and its fundamental role in biological processes. Through time, his work got him to define a new chemistry, which would be supramolecular chemistry. It works on complex entities that come from the bond of at least two different species, chemical species that are bound together in a non-covalent manner, different from those that are created from atoms in a covalent manner. So in this new field, he developed the self-organization processes chemistry as well as the adaptive chemistry. Professor Lehn belongs to different institutions, and he has been awarded different international awards. Moreover, he's a wonderful speaker. His speeches in public from them, I will highlight the way in which he transfers ideas and he explains the unknown things of science to many. The University of the Basque Country firmly believes that the generation of knowledge demands the broadening of knowledge. So we have the will to be able to transmit this, and we do this with the Excellence Campus and our PhD. And we do this together with the Donostia International Physics Center that is led by Pedro Miguel Echenique and that has fostered, together with the support of Telefónica, and I would like to thank them, the organization of this conference. The evolution of the universe has created a matter that is more and more complex, from the atoms to the living organisms or thinking organisms. So as a consequence of their interaction of molecules, we have more complex matters. Therefore, chemistry establishes different sources, amongst them molecular structures and living structures, and the advanced chemical and the advanced chemistry allows us to build with molecules that are more and more complex until we reach the supramolecular level. With the interaction of molecules, we can control molecular processes. This demands the use of information in molecular programming. Chemistry is a universe in itself. And I'm sure that this is going to be a very interesting talk for each and every one of us so that we're able to understand these concepts in a better manner. So the floor is yours, Professor Lehn.
Sorry not to speak Spanish. Buenas tardes, nevertheless. So, it's for me an enormous pleasure to be here, being able to give a lecture in this fantastic building. If I had not been a chemist, one of the things I would have liked to do is to be an architect. And you will see, perhaps, in my lecture, the relationship which can exist, nevertheless, between chemistry and architecture. This is a building where we have surrounded by art, but the building in itself is a piece of art. I knew Bilbao before the building existed. It was a totally different city. Now, Bilbao is on the map. And this is due to art. And art implies, especially in this case, also science. You cannot build this without having enough metallurgy, without having enough sciences, material sciences. So it's a sort of a symbolic building, not just for art, but for human culture, which also contains, of course, science. I would like to thank Pedro Echenique and those who have invited me to be here today. Also, the director of this institute, of this museum, of the institution, because as I said, it's really quite, for me, a great pleasure to be able to be here. I had a good friend, Claude Vasconi, who was also an architect, and he had a project for the fair in Sevilla. You know, there was a world fair, and he asked me to be sort of advising to it. And on the project, I was called molecular architect as an advisor. I liked that very much. So, we are now on a trip through science and knowledge. Again, here I would like to point out this fantastic work done in San Sebastian. These meetings called Passion for Knowledge. I think, yes, passion for knowledge is something which can drive you. It drives me. It drives many people. I'm sure it drives also many of you. That's why you are here, I hope. And I hope to be able to tell you a little bit about what has been going on and to place my field, what we have been doing, in a much broader surrounding, much broader panorama. The title I have given is From Matter to Life. This is used, that is like an advertisement in a newspaper, you know. Sounds very exciting, from matter to life. Up to there, it's fine. Chemistry, then people begin to be dubious. Chemistry, really? What does it have to do with it? The answer is, of course, chemistry. But in order to show this panorama and to have really a trip through some parts of science, we have to start very, very early. We have to start here, at the start of the universe. The universe started where is this? This is something which works. There was one which worked. It started with a Big Bang, as cosmologists tell us. At that time, there was no chemistry. Let's say there was energy. But it cooled down very quickly, and progressively, particles formed. And so, very quickly, 1 second, we are here. Particles are forming, and this is the age of physics. No chemistry. The laws of the universe, the physical laws of the universe, are running the show. It cooled down more and more. Particles formed atoms. Atoms connected to make molecules. Chemistry started about here. These molecules, in other words, the connections between atoms, building larger architectures. You see the word again, architecture, we'll hear it more. Built these molecules, which became, by some processes, which we will see a little bit about, more and more complex. They associated together. They began to define regions, as aggregations, regions where there were compartments, and these compartments sort of made an inside and outside and evolved progressively towards what we call living cells and life. And then biology started on our planet about here. That's the age of biology. But it didn't stop, at least on our planet. Evolution continued, and out of the initial primitive types of life, more and more complex forms of life evolved, multicellular organisms, further and further, ending up on our planet in an organism, the one sitting in this room, and with a property, thinking, the most fantastic property we know. It's represented here by this sculpture of Rodin, the thinker. Now, before I continue, I would like to make here a remark. At the end of this picture is sitting this person. But do you think it's finished? No. Evolution will continue. We as human beings are only a point in space and time. It will continue. If people think they can stop it, impossible. It will continue. And if we don't continue on another planet, it will continue. I am convinced there is life in many other planets. Many planets are found. The universe is enormous, with enormous number of planets. So, this is the evolution from matter to life. Now, cosmologists tell us that there is only 5% of visible matter in the universe. So we have still a lot to learn, because this is the matter that matters, but there is 68% of dark energy. Numbers fluctuate a little bit from time to time. 27% of dark matter, which means what? 95% of darkness. 95% we still don't know much about. But it is not our purpose today. We are interested in it, but it doesn't concern us really. We are really concerned about this matter, the 5%. So if you look at the evolution of the universe, we can try to define it by some parameters. I have chosen as parameters complexity and information as a function of time. Matter was first divided. Then it became condensed. Molecules got together, making larger aggregates. But it's not enough. It must organize. So it became organized in some way. Then it became living in some way. Then thinking. And beyond, something difficult to think about our own thinking. But we can't exclude there is something more complex than our own thinking. Maybe in 1,000 years, if we can come back, we will know about. So, matter seems to be evolving, at least in some regions of the universe, under the pressure of complexity, information, more complex structures. And this is the picture which one can try or wants to try to understand. So then there is a big question one asks. Maybe not necessarily the most important one, because there are physical theories, quantum physics, general relativity, which are of course very important. But the big question for us is, how does matter become complex? How could the universe generate an entity, a human being, capable of inventing these explanations of the origin of the universe, capable of generating Planck, of generating Einstein? This is really very basic for us. If there is no Planck, no Einstein, there is no explanation. So, how does matter become complex? From the elementary particle to the thinking organism. And maybe, as I already evoked, there may be more complex forms of matter even. So for doing that, mankind, in its curiosity, had this fantastic idea of inventing science, which has, of course, many fields, but let's look at three main pillars. Physics deals with the basic laws of the universe. Everything depends on that. There is no chemistry, no biology, no life, no thinking without the basic laws of the universe. This being said, we want to understand also the rules of life. These are not laws, these are rules of life. And so, to make the connection between the two, the bridge is built by chemistry. The bridge between general laws and specific expressions of those laws. Why do we look the way we look? What is the way in which atoms, molecules evolve progressively to give a thinking organism? And this is the mission of chemistry, to try to build that bridge. Now, is there an answer to that basic question? Yes, there is one. So you say, what is he telling us? This is stupid. Because if we knew the answer, we would have it in all papers. El País would have big titles. The answer is this. Yeah, we know the answer. But we know the answer, this path towards complex matter, just by a word. And this word is self-organization. It means everything and nothing. It means it happened by itself on the basis of the laws of our universe. But it doesn't tell us how it happened, what are the steps we went through. Now, if we admit that the evolution of the universe is based, the complexification of matter is due to self-organization, we must admit that in our universe, it is a cosmic imperative. In other words, this would mean life as we know it is not an accident. Life is built in the structure of our universe. And there have been a number of scientists, for instance, Christian de Duve, who wrote a book called Vital Dust, which tries also to promote the same idea that self-organization is an inbuilt property of our universe, and that's why we are here. Now, self-organization, we still, because our friends, the physicists, have not yet come up with a unique theory, we still have to look at it in two ways. One is self-organization on the large scale, cosmic self-organization of the universe. And people tell us, cosmologists tell us, that after the Big Bang, there were inhomogeneities in density of matter and in rates of expansion. And gravitational forces acting on this led to the formation of galaxies, stars, planets, and so on. This is very important, but it's not our matter today. On the other hand, there's also a self-organization which rests on the getting together of the bricks and the elements constituting the universe through what one may call electromagnetic forces. And there, what happens is that a sort of a random combination of these bricks and pieces generating these structures, different structures, and some a bit better meaning in terms of the physical conditions, thermodynamically, some are less good, and so the thing evolved without any finality. There is no need for a finality. It just happened, and then it evolved in some way or another. Now, we may at this stage already consider that before there was any evolution of biologically living organisms, the Darwinian evolution, there must have been an evolution which had nothing to do with Darwinian and which is much broader. An evolution of matter, non-living at the beginning, towards the threshold of life. And this was the evolution, this was built in again, even without any life. There must have been an evolution, because it's difficult to imagine that you jump from a particle to a cell, a bacterium or something like that. So there was some kind of a process which led to the organization of matter progressively going to the threshold. And that is a very exciting field in which we are now interested to see what are the possibilities, what are the pressures, what are the factors which may lead to a more and more complex form of matter even before it is living. Of course, then at some stage, we still have a black box. We don't understand yet the jump from non-living to living matter. But it gets closer and closer, and the gap between the two gets more and more restricted. We can maybe talk about this a little bit later. So within this concert of science, this orchestra of scientists, what is chemistry doing? Chemistry is concerned with the structure of matter, how it is built up, and the transformation of matter. How can you convert given pieces of matter, we'll see what these pieces are in a moment, into others? This is something which attracted me to chemistry. Transformation of matter, this gives you, it's a sort of a power trip, you may say, when you can tell yourself, I am able to transform matter, to take things and transform them into something else. This is really, I think we will see at the end, has something to do with art also, this creative power. So chemistry has these two properties, trying to understand the structure of matter, trying to understand the rules of the transformation of matter. But there's a third one, which will come up a bit later. Now, let's have a look through a little bit, just a little bit, very small aspects of history of, let's say, science, chemistry. About 2,500 years ago, there was a part in Europe, Greece, where there were philosophers. Philosophers at that time were everything. There were scientists, there were, at that time you could be everything. And there was someone called Empedocles who proposed that matter was made of 4 elements: fire, air, water, and earth. And the combination of these elements made properties. Fire and air was hot, air and water was humid, water and earth cold, earth and fire dry. Okay, it's a try, it's simplistic, but it's a try. However, at the same time, somebody else went much deeper, and this was Democritus. Democritus said matter is particulate. It's formed of particles which cannot be made smaller, which cannot be cut more. Atomos, atoms. Now, this, of course, doesn't mean that Democritus discovered atoms, because in order to discover something, you have to prove that you are right. An idea is nice, but in science, you have to prove. But now, I want to make you understand that you cannot go around the city and convince anybody that matter is made of particles. Do the following. Take a glass of water, take a piece of sugar, Put it in a glass of water. What happens? It disappears in the glass of water. We see it dissolves. What does it mean? That obviously this glass of water and this piece of sugar must be particles, otherwise it couldn't mix. And it is said, although one doesn't know much about what Democritus wrote because it has disappeared, but his disciples, it is said that on the basis of this kind of things that Democritus came to the idea that matter is particulate. If that's true, it's fantastic. Simple observation, fantastic consequences. I would like to know if Democritus thought this way. He must have had some ideas. Now, let's jump. Maybe make big jumps. If you want to study matter, which is a very complex thing, one way, what we have to start with is to separate something very complex into smaller pieces. And for chemists, that means analyzing matter. And among the processes, one is to make crystals, another one is to distill, and we all know that distillation is a very important technique. That's the way we make brandy, and so on. And I just show you here a book which I like very much because it was a sort of a real first book in chemistry, written by Hieronymus Brunswick, Liber de Arte Distillandi de Compositis, which was printed in 1512 by Jakob Grüninger. And this was the Compositis. The first one was Liber de Arte Distillandi de Simplicibus, das Buch der rechten Kunst zu destillieren, das einzige Ding. And this was also printed in 1500. In fact, as you may know, Gutenberg was sometime in Strasbourg and he invented printing. And this in Basel was also a center for printing. And Grüninger was a printer in Strasbourg, the city I'm coming from. Now, it's quite nice for those who are chemists here, because you see these two guys, of course, don't look like somebody now in the lab. They have another type of outfit. But this kind, you know, for those who are chemists, that's a fractional distillation, 1500. Not so bad. So, at that time also, theory began to be more and more supported, or the need for experiments became more and more pregnant, more and more strong. And among those people who promoted the necessity for supporting thoughts by experiments, there was this man called Paracelsus, with a nice name, Aureolus Philippus Theophrastus Bombastus von Hohenheim. That's quite something, huh? If you sign your letter this way, it takes you half an hour to sign it. He was born in Switzerland, in Einsiedeln, and he died in Salzburg. He said, nothing is a poison, everything is in the quantity. This is one of his. Of course, Paracelsus was a very, somebody who was not easy going along with people. In Basel, he burned books he didn't believe in publicly, and of course, he was thrown out of the city. Then he was a citizen of the city of Strasbourg, and he had also to leave because he was a bit not so getting along very well with the people in the city. Let me just show you one thing. Paracelsus, he is buried in Salzburg. This is his tombstone in the San Sebastian Cemetery in Salzburg. Why is this cemetery interesting? First of all, not many chemists have this kind of tombstone, but there's something else there. The family of Mozart is also buried there. Not Mozart himself, because he was in a public grave. But here is Constanze Mozart, geboren Weber, and the other family of Mozart. So if you go to Salzburg, in addition to listening to nice music, you can also see this and pay also some respect to Paracelsus. Now, with this analysis, need for experiment, one progressively established that there were pieces in matter which got names. And just an example among many I could have taken, in 1718, there was this kind of table was proposed with nice symbols. Well, they look nice, huh? And therefore it is copper, iron, lead, tin, zinc, gold, silver, and so on and so on. Okay, was a try to organize what one had learned about composition of matter. But it looked not, people were not very happy because there was no rule for that. So, was there some order? Was there some rule? Was there a reason to put it in a given way? In the middle of the 19th century, the situation was really terrible. People didn't really know so much how to organize that. But, as you know, often in science, flowers emerged. And the one who got the prize, if I may say, is this one, Dmitri Mendeleev. In 1869, he wrote one of the papers which for me is one of the most important papers in the whole of science. 1869, on the relationships between the properties and the atomic weight of the elements. He had noticed that there are regularities if you look at the properties and the atomic weight of the elements. This was published in Zeitschrift für Chemie, 1869, and he organized the elements known at that time in this way, in columns and rows, according to their properties. He also noticed, and this was the next step, one thing, you have to organize it. The second is to use this organization for making predictions. And there were places where there was nothing. And he said, he put question marks. He said, someday we will find it. And of course, they were found. So not only did Mendeleev propose an organization for the pieces which make up matter, but he also predicted that others will be found. This is now called the periodic table of the elements, and the present-day aspect is this one. These are the bricks of matter. Now, again, this is a consequence of that paper of Mendeleev, but it is one of the most fantastic advances of human knowledge. Why? Because that table shows you all the bricks which make up visible matter in all the universe. Now that sounds a bit terrible because it sounds dogmatic. And the scientist should not be dogmatic. But it is full, this table. It goes from hydrogen to helium, lithium, barium, and so on and so on and so on. Now, why is it full? Because atoms are made of a nucleus and electrons, and there are protons in the nucleus and electrons running around. And what do? You count. 1 proton, 1 electron, 2 protons, 2 electrons, 3 protons, and so on and so on and so on. And between 1 and 2, 2 and 3, so on and so forth, there's nothing else. So it is complete because it's arithmetic, simply. So this tells you, you can look at it, it tells you that if there is a living organism 1 million light years away, it will have the same atoms. You realize what that means? I find it unbelievable that sitting in this room, sitting on this planet, we're able to say that visible matter in our universe, from the planet Earth to the end of the universe, will be made of hydrogen, will be made of helium, will be made of lithium or barium, boron, and so on. I usually recommend that you take a nice picture of the table, put it over your bed, and look at it every morning. Because it gives you confidence that things are possible. It's confidence. Sometimes you need confidence. You need to build up your confidence. And when you say, okay, we have been able to know how things are built up. Okay, let's continue. So this is the playground of chemistry. So chemists are like children. They play Lego. They have Lego sets, they have pieces, and these are the pieces. And with that, you open the world of all the combinations. Now, because it's a place where there is art, let me also have some pieces of art in my talk. A good friend of Mendeleev was another very good chemist, Alexander Borodin. Now, Borodin is much better known now as a composer, but he was a very good chemist. There were 2 reactions for the chemists in the room, which are discovered by Borodin much before the others, what is now called the Hunstiger reaction, but it's Borodin who found it, and the other one which Wurtz found, but it's Borodin who was the first one. Of course, he wrote The Prince, Igor. This is certainly by you much more known than the rest, symphonies, quartets, and so on. He also considered himself more as a chemist than a composer. He said, composing, I do that on the weekends when I have the time. Now, structure of matter, the elements. Then you have to transform it. You have to know how, when you mix things, what else can result from it. And there is a very important rule, the rule which tells you how things which you mix can rearrange to others. And this is due the first time this was sort of clearly proposed, the chemical reaction, was by Lavoisier when he said, rien ne se perd, rien ne se crée, tout se transforme. Nothing is lost, nothing is created, everything is transformed. This is, without this, chemists can't work, because it tells you that the elements you have, you mix them, they rearrange, and they make other combinations, but the elements remain the same. Now, we have talked about the nature of elements, the transformation, the rearrangements, and then came up the idea that maybe these things are connected, and these connections make the entities which, again, in the middle of the 19th century, one began to call molecules, combination of atoms to make architectures which are molecules. And people began to try to make a graphic representation. A graphical representation, many were proposed. I show here just three: Wurtz, Kekulé, Wurtz in Paris, Kekulé in Bonn, and Loschmidt. Now, Wurtz and Kekulé are fine, it's not wrong, but they are quite far from what you do today. In fact, Loschmidt in Austria, he was much more advanced. He represented big circles. He used big circles to represent carbon, small circles to represent hydrogen. A double circle here was oxygen. And to look at one of them, that's the way we write it now. If you replace a small circle by H, a big circle by C, a double circle by O, you get ethanol. That's exactly the way we write it now. Just to say that it began to take a shape in 1860s, in the middle of the 19th century. It began to be a shape and began to have reasonable ideas about what molecules were. So, this is a graph. This is a definition of connectivities. But is it just connectivities? Is it just a graph? Is it not more? In 1874, independently, two persons, Van Toff in the Netherlands and Lebel in France, proposed that, in fact, molecules had a shape, an architecture. They were not just graphs, but they had a shape. They had angles, they had lengths between the connections, and so on. This was an independent proposal, and of course Van Toff did much more, and he was the first Nobel Prize winner in chemistry. So we now are at a stage where we know that these bricks which make up matter, atoms, they are connected, and these connections give them a shape. Since we talk about matter and life, I must mention somebody, Pasteur, because he was the first one who did many other things. But one very important thing for chemistry and for matter was to realize that some molecules look like a left hand or a right hand. And this is, and in biology, in fact, it's either one or the other. They're usually not mixed. And since in Greek, hand is kairos, so this was called molecular chirality, the property that some molecules are left, like a left hand, and the other ones like a right hand. And this is the relationship with the plane between the two. So, this was a very, very quick run through history from the atom to the molecule. And this is the build-up of molecular chemistry. This molecular chemistry which uses the bricks, the atoms, and links them together by a strong linkage. Chemists call that a covalent bond, but forget about the name. It's just a strong bond which holds things together. Two milestones in this development. One I would like to mention is the production in the laboratory of urea by Friedrich Wöhler in 1828. this was a very important thing because Wöhler made urea from a substance chemists call ammonium cyanate. Ammonium cyanate is considered as a mineral. Urea is contained in living organisms, in urine. And this led not only to the realization and to the result that indeed it was possible to make urea from ammonium cyanate, but at that time there was still this thinking that you need a magic force to produce anything which is contained in a living organism, vital force, in order to be able to make it. By making urea from ammonium cyanate, Wöhler changed that. He had I would say, a metaphysical, a philosophical effect, because he showed there's no need for any vital force. The molecules which are in organisms are molecules like others. They are molecules. Now, molecular chemistry then built up by many, many, many people who worked on it, many chemists. And just to give another example of now a complex object, it was mentioned already in the introduction, vitamin B12 was, which is a vitamin we have in our body, very important one, a very complex one. This is now a very big molecule compared to urea. This was made in the laboratory by what chemists call synthesis, putting things together, the atoms, in many steps by two groups directed by Robert Burns Woodward at Harvard and Albert Eschenmoser at the Polytechnical School in Zurich. They had over 10 years or so co-workers, and altogether it was something like 120, 150 man and woman years. As it was, I think, mentioned, I worked with Woodward, and this is my piece. Now, yeah, it seems funny, but you know, this is a good illustration of what science is. You are part of an enormous project as a scientist. You bring a stone to it, it's your stone. Nobody takes you away, take it away. This is my stone there. And of course, the other stones have been produced by other people. So you are in an enormous enterprise where each one helps each other to build up, in this case, a molecule, but in more general terms, the scientific passion for knowledge. Now, that was molecular chemistry. I should say that, of course, vitamin B12 represents a step in the 1970s, and since then, much more has been done. More new reactions have been found. New, more complicated molecules have been made. New materials, new drugs. And so molecular chemistry is very alive. It's an adult science, but it's a very alive one, and it continues. And nowadays, one probably would not make vitamin B12 anymore by the 90 or so steps, 90 steps. You go from the beginning to the end. That was quite a Himalaya of the chemical synthesis. But one wouldn't make it exactly the same way anymore because new reactions have been found which are more efficient, which allow you to do it in another way, maybe reducing the number of steps and all that. But you can come to the point that you ask the question, okay, we now know quite well how to make complicated molecules. But is there something else we should think about? What you see here is a cancer cell, the blue sphere, and two killer cells. What are the killer cells doing? As you are sitting here, the killer cells in your organism are running around, trying to find out the cells which have gone wrong, and their mission is to destroy them. If you don't destroy them, the cancer cell may develop. So the immune system, the killer cells of our immune system, does the job of cleaning up. How do they know the other guy is a cancer cell? They shouldn't make a mistake. If they don't destroy a transformed cancer cell, you have a problem. If they destroy a healthy cell, you also have a problem. So something must tell the killer cells that the other one is indeed the cell to be destroyed. Another example, these blue dots are the HIV virus, which when they enter in collision with a white blood cell can infect How does the virus particle know that the other one is the target and can now be infected? How is it? Now, I suppose you realize that these objects are defined by a membrane that gives them the shape. This membrane, this one and this one, are made of molecules. It's like a sort of a soap bubble which children use. And in these soap bubbles, there are sticking molecules called proteins, which are, so to say, the hands which can give a feel to the killer cells touching the other one and giving information about the fact that the other one is either a healthy or a transformed cell. So there is a chemistry which happens between the objects, not inside. Inside the objects is molecular chemistry. Between the objects is a chemistry which deals with, let's say, societies of molecules, assemblies of molecules, what molecules do when they get together. So this may be termed chemistry beyond the molecule, which is supramolecular, beyond molecular chemistry. And here again, now, in order to be able to handle these objects which get together, one has to try to control the forces which hold them together, which chemists have called non-covalent bonds, but again, forget about the term, it's not important. It just means that between the molecules formed by the connection of atoms, there may be connections between the molecules of a weaker type, which lead to these larger entities and which make the molecules able to attach to each other. The basic features of this process is recognition between the molecules, the way they can react with each other, and the way they can carry each other, for instance, through a cell membrane. In our body, the membrane of the cell regulates what gets in and out, and these processes which allow things to get in and out are transport processes. But the most basic process is this one. How do molecules recognize each other? How do they do it? Molecular recognition requires, first of all, binding. If they don't interact, they ignore each other, so nothing happens. So they have to have some way to interact, to get a feel for each other. But this is not enough. When you want to recognize somebody in the street, what do you need? You need information about the person. So, molecular recognition, the way molecules recognize each other, is an information process. Otherwise, of course, it couldn't operate. It's, you may call it molecular information. I don't want to elaborate more on it, except for saying that there's a simple way to understand what it can be. It can be considered as a complementarity of two features, the geometry of the object and the way it interacts. You may know that a plus charge attracts a minus charge and a plus repels a plus. Of course, things are more complicated, but that's the basis. And in the 19th century, where I didn't talk yet about these things, there was nevertheless a deep understanding already about what this can be. This is due to Emil Fischer, one of the greatest organic chemists who ever existed, Schloss und Schlüssel. Molecules have to fit together like a lock and a key. The lock and the key is the complementarity, and in addition, even more, if you have the right key, you can open the door. So you can also induce a function. This was fundamental. At that time, it wasn't called molecular recognition, but lock and key. And this was good enough. Here is Emil, Schloss und Schlüssel, a paper which he wrote in 1894. He got his PhD in 1874 in our university. I make a big, some publicity for our university also. But I think it's an honest game. At that time, Europe has changed over the years. And the French had lost the Franco-Prussian War. So Alsace had become German. For science, it was pretty good for us because we got a lot of fantastic scientists, especially chemists. And the professor with whom Emil Fischer was Adolf von Baeyer. Emil Fischer was the second Nobel Prize. Bayer was, I don't remember, the third or fourth, or no, a bit later. And a lot of people, very, very good chemists, came around. There were also physicists. Röntgen was a professor in Strasbourg, but he didn't find X-rays in Strasbourg. He found it in Germany. In the other part of Germany, because we were Germans at that time. So, let's now have a look at molecular information. The most important information system for us, human beings, is the information stored in our genome. The information which makes us a human being and not a tomato, or an elephant and not a bacterium. What is it? Let's have a quick look. The information which writes our genome is a long string, a chemical string, on which are attached letters, only 4 letters. 4 chemical groups, this one, this one, this one, and this one. Chemists have given a name to that. You know, chemists like to do that. They give a name, which is more and more exotic. Adenine, guanine, thymine, and cytosine. AGTC. These are perfectly well-defined chemical groups, rather simple ones. But this is enough to write the genome of all living organisms. It's a sequence, a very long sequence of these letters in a given order, G, G, A, T, C, whatever, no? And 2 of them wrap around each other and they make the famous double helix, of which you have a model there. So, we are the result of a compilation and combination of 4 chemical letters. If you look a bit further into one of the letters, let's look at adenine. What is adenine? This guy here. Adenine is building together, combining together 5-HCN, hydrocyanic acid. You can, for instance, see HCN, HCN. Others have a bit rearranged, but there are 5 HCN. HCN exists in interstellar space. So, it's easy to imagine that you can make adenine out of 5 of them when 5 get together. If you add a little bit of water, you get the second letter, guanine. Some more transformation, and you get the other 2. In other words, there's a very simple chemistry which can occur in space and which gives you the 4 molecules, molecular groups, which write the genome. So you see what we are? It's sort of, we have to be modest because we are the result of a combination of these 4 letters. But then the question is, okay, in this strand, this is the writing of the information. The sequence gives you, this is a chain on this thing, stick on it. This is the sequence. But you have now to read that information. How do you read it? You read it now by a supramolecular way. In other words, the chemical letters are molecular, and the interaction between the letters are of the supramolecular level, and the little dots here represent these weak interactions. The lines, the continuous lines here, represent the groups. For instance, oxygen is painted in red, nitrogen is painted in blue, black carbon, you don't see it too well, but these are. And then they interact by either 2 connections between adenine and thymine, or 3 connections between guanine and cytosine, which means the following. It's a binary system. The information in our genome is read by 232323, like a computer does 010101. Very simple. Why so simple? Because usually evolution stopped when it was good enough. Of course, you can imagine more complicated things. You can imagine there are 20 letters. You can imagine that there are more interactions. But 4 letters pairing 2 by 2, that's enough. And possibly that's why it was stopped, or why evolution didn't go further, was enough. Because now some chemists are trying to make other letters. That's a bit a complicated thing, but there are people who have been introducing other letters in genomes and have reading processes. So I insist here again on this fact that chemistry is, of course, the science of the structure and transformation of matter, but it's also an information science. It's a science of informed matter. Any chemical process, and especially the biological ones occurring in your organism, is an information process. As you are sitting here, your molecules all obey molecular recognition because they have to find the partner, they have to do things which are very well defined, which occur in given regions of the cell, at a given rate, talking to each other and all that. So it's a very complicated process, but it's a basic process which makes chemistry not only the science of the structure and transformation of matter, and I like to insist usually on the fact that it's an information science. The storage of this information is the molecule, like the strand in DNA, and the processing, the reading, is supramolecular. So let's just give a very simple example of how you can play with molecular recognition, how you can try to understand it. If you play with locks and keys, if these are 3 keys, that's a lock. Of course, the red key is the one which fits. So when we started our work, I was interested in, you will see in a moment, in simple things. You start simply. So in science, often it's the case. When you have a complex problem, you should first simplify it, because otherwise you get into trouble. So you simplify, and then you rebuild complexity. So what are the simplest keys in chemistry? Let's say even just in geometry. The simplest objects are spheres. And they are spheres in the chemical world, in the Mendeleev periodic table of the elements. One class is called the alkali metals, which contain notably sodium, and sodium, all of you know, kitchen salt is sodium chloride. And this is a very nice collection of balls with one charge and getting bigger and bigger, from lithium to sodium to potassium to rubidium to cesium. Nice collection. So you can ask yourself the question, how can I recognize one of those in the collection of all of them? I was especially interested in that because I was really, in fact, interested in the chemistry of the neural system, neurochemistry. And so what does it have to do with that? What it has to do is that 2 of these ions, sodium and potassium, carry the nerve influx, which makes neural electricity traveling along the nerve. It's a change of concentration of sodium and potassium ions across the nerve membranes, which makes what is called the action potential, which propagates the information along the nerve. And for a chemist, I thought at that time, it was many years ago now, 50 years or so, I thought, okay, I'm a chemist. That's a way for a chemist to sort of try to approach the problem. But we were very quickly sort of driven, like, you know, in science, often in research, you have a goal, but you see interesting things opening up, and then you have to decide, do I continue the initial goal or do I go to these other things? And the decision was taken to try to understand, on the basis of these simple objects, to try to understand molecular recognition. So the question was, how can you selectively bind, catch, so to say, and carry those things through a barrier like a membrane? And this is spherical molecular recognition. So many years ago, in 1968, we made cages, molecules, in the laboratory by the normal laboratory techniques we chemists use, which had a cavity. This cavity is, of course, the lock, so to say. So these molecules are molecular locks, and the design is such that you can change the size of the cavity so that this one fits best with lithium, this one fits best with sodium, and this one fits best with potassium. And this was, for us, the beginning of studying molecular recognition in the simplest case, spherical recognition. Of course, the molecules in the membrane of the nerve, which do the same job of distinguishing sodium from potassium, if they can't distinguish, of course, you have a short circuit. That's not good for you. Those are much bigger ones, and they are proteins, big molecules, which have to do many other things. But the simple process, analyzed, cut out of the complicated one, is to make simply locks for keys, or sometimes keys for locks. I come back to that. So, again, this is one of those. You see, that's the potassium one, this one, the cavity, which is just right for potassium. That's another representation. Why do I show you that? Because there's a chemist, Bela Visi, at the University of Veszprém in Hungary who has turned sculptor. And he made for me, I was very happy about that, this very nice sculpture, which is one of those cryptates, which we call them, because the molecules are crypts. When you put something in, we call it cryptate. And this is the one with potassium. Now, Bellavisi is, in fact, somebody who has a lot of imagination. I show you 4 more of his sculptures. I will only discuss 2 of them. But he made quite a lot, and you can find him on Google. You put Bellavisi on Google, and you find many of the things he has done. Let's just show you a few more. This thing here. This is, the name is the scent of the rose. So why is it the scent of a rose? There may be chemists in the audience who know what is the scent of a rose. It's a compound called phenyl ethyl alcohol. It's like ethanol, the alcohol we know, but you replace one H by another group. And look at the sculpture. This is the phenyl. This is one carbon, another carbon, OH. So it's very nice. It's a sort of taking a molecule and making a scent out of it. It's not bad, huh? And I showed it to artists who know nothing about chemistry. They found it's a very elegant way of expressing it. These 2 are called the dancers. There are 2 molecules found in nature, linalool and linalool acetate. They have these groups here, dance. They have the arms lifting there. And some others here. He has made a lot of things which are very interesting type of pictures. So you see, you can make pieces of art just by looking at the structure, the architecture of molecules. So, applications. We are often asked, yes, but what is it good for? My opinion is, at the beginning, it's good for knowledge. But if you have knowledge, you can have applications. Without knowledge, you have not much of an application. So first of all, the game of locks for keys and keys for locks is very important for drug discovery. Because what is a drug? A drug is a key for a biological lock. You take a drug, it should in principle be designed to be as selective as possible to look for the biological entity which it will either close, inhibit, or open, activate. I will not go further into that because I think it's quite clear that a drug, a pharmaceutical product, is a key for a biological target lock. One can also try to think to make medical diagnostics. A lot of work has been done in this area to make medical diagnostics, to understand how you can transfer a drug into a cell and so on. We have been working, we give you an example, gene transfer and biomaterials. I just want to illustrate that briefly. We had, among these cryptans and cryptates we made many derivatives because we wanted to sort of explore this area. And we made one which has this type of structure into which one could introduce one of these elements, which is the periodic table, europium. And it happens that when it goes in there, it has a red luminescence. And you can attach this little nanobulb, a sort of a bulb, you can attach to a protein, an immunoprotein, and make a system for medical diagnosis. And this is the machine which has been built thanks to the hard work of Gérard Matisse. He's the one who really got it going because we studied the basis and he had seen that there's an application. And so they developed, after many years, a machine now, this medical diagnostic type of entity, which is used now in a number of hospitals. What about transport processes? I told you that one of the features of interest is that the molecule binds another one and helps it to do something, for instance, to go through a membrane. Now, there's a field which is developing. It takes time. It's not an easy one, whereby you try to take a piece of DNA for a defective gene and help it to go through a cell membrane. Normally, the piece of DNA is, so to say, very water-soluble. A membrane, as I said, is like a soap bubble. So this piece of DNA wants to stay in the water. It doesn't want to go through the membrane in the cell. So you make a sort of a stealth type of object, a synthetic vector, which wraps around the piece of DNA, hides all this from the outside, and makes it able to cross the cell membrane. Then it goes to the nucleus, it gives and undergoes a number of transformations. Eventually, it produces the protein, the molecule which is lacking, which the defective gene could not produce. One application in which we have been working with one of my brothers, who is a cell biologist, Marie Lehn, is cystic fibrosis. Cystic fibrosis is a disease where the lungs, there's a molecule, a chloride channel, which doesn't function well in the lungs. And then mucus accumulates, and usually you cannot breathe anymore, and you die. There are now some drugs coming around. One way is to try to introduce the correct gene, which makes that channel, which makes the thing function again. And of course, since the airways in our lungs are accessible from the outside just by inhaling, then you can hope to correct for this defective gene by transfecting, making a genetically modified entity. It's very good to do genetic modifications. I can back to that if you want questions. I'm ready to take up questions for that. I have a strong opinion about it. So you can use it for gene therapy and for biotechnology, introducing genes into organisms to make genetically modified organisms, and for gene therapy to cross the membrane and correct for defective genes. In fact, there was something very important also done by a lab in Paris, where bubble children are children which have no immune system. So you put them in a bubble so they can isolate it completely from the outside. And there's now, people have been able to, have a beginning of using transfection to correct the immune system. It's a bit more complicated than that, but it's just to give you an idea. What about materials? We had made over the years, among the many things which one can do, polymers. What is a polymer? This is a polymer. Huh? It's nylon is a polymer. Plastic materials. These polymers are made by linking together molecules by strong bonds and making long, long, long fibers. That's nylon and many others. We had developed materials where the units which go together and make the long fibers are connected not by the strong so-called covalent bonds, but by the weak so-called non-covalent supramolecular bonds. This was published in 1990. And I usually want to cite this example when there are people in the audience who have the finances in their hands. Why? So in 1990, we published the concept. It developed a lot. Many people worked on it. And the field developed a lot. In October 2013, I got a message from the CEO, the chief of a small company, who told me in the message, we have used supramolecular polymers as biocompatible polymers to make cardiovascular implants. Because since the connections are weaker, the hope is, and the idea was, that since these things are less stable, if you can make an implant, it will with time be resolved and disappear. This is especially true for children because they are very active, so there are many cells coming in and they can invade the structures which are formed. Furthermore, children grow. So if you make an implant, the implant shouldn't prevent the growth of the heart. And supramolecular polymers being so it's more extendable were thought to allow that. And there are children having a severe congenital cardiac malformation which require cardiac reconstruction. Where do we stand? Here is Dominika, which was implanted by Leo Bokeria. She was 4-year-old, and in October of 2013, Leo Bokeria is I didn't know him, of course, but he's a very well-known pediatric cardiac surgeon in the Bakulev Center in Moscow. He used these implants made by the company called Xeltis. And here is Dominika. This was the first checkup 3 months after implantation. It's now October 2013. It's a year and a half. She's fine. She has no problems. 10 other children have been implanted since then, and they're all fine. So, the point I want to make is, how could we have predicted that someday supramolecular polymers will end up in the heart of a child? Impossible. So you do it because you find it's interesting, it's curiosity, you want to do something new, and there are enough intelligent people around the world who see what one can do with it. This is my couplet for basic research. Knowledge. Knowledge can be used. Without knowledge, no use, nothing. So I'm, of course, very happy about this, that you see that, okay, something which is it also applications in material science and so on, but especially here, it's something where, okay, you have helped people. So 10 children have now been treated. And they consider it as a real breakthrough in surgical practice. So let's go back to the main theme, towards complex matter, towards life. When you understand a bit more about molecular recognition, how molecules get together, you may think of using this knowledge to let the system build itself up. In other words, from pieces, they recognize each other, and they will be able to build something. When you want to build a house, you have to take the bricks and put them by hand next to one another. Here it's like as if the bricks had some way to feel each other, to recognize each other. For instance, this building has no bricks, so it doesn't work here. But some other building, the Colosseum in Rome, for instance, where the bricks would have information and it would just get together in the right position. This is then a way in which you can use the recognition between the objects to generate, by self-organization, molecular recognition directed, spontaneous but controlled generation of complex architectures. To illustrate that, let's take a biological example, a virus. I could have taken many viruses, but this is one of the first ones, if not the first one, which was understood. A virus of the tobacco mosaic virus. It is not living. A virus doesn't live. A virus is dead when it is not in a cell. It begins to reproduce itself when it goes into the cell. But here, what we will first have a look at, how does it build up? The construction of this virus is 2,130 bricks, which get together with the RNA, the genome of the virus, and the bricks are built in such a way that the surface-to-surface contact, in other words, one molecule which has a site which is complementary to the other one, like this. So they can attach to each other. You can either have recognition processes between two different molecules or the same molecule recognizing another part of itself. And this is what happens here. This is a more detailed chemical structure of this virus. These pieces, these proteins, they bind surface to surface. And, since they are built like a piece of pie, sort of, they are a little bit conical, like so triangular, they must necessarily, when they attach in this way, they must go around a circle. And it's not just a circle, because it can form a helix, and then it builds up the final virus. Now, this looks like magic. It has absolutely nothing magic. If we want to understand, and people have done so, all the features here are chemistry which you understand. It's organic chemistry, it's structural chemistry, it's interaction between molecules. That's the way it is. Now, nevertheless, it seems sort of really quite magic. And then you can find on Google a video of people, I think from California, who have done the following. I must tell you that. It will be a little bit longer, but I guess these stories may be interesting. There are some viruses which are like spheres, composed of caps, which make a sphere. And it's known, some viruses are known to have so many of those things. So these people, they took pieces of plastic, so it's macroscopic, it's centimeters. And at the places where one knows that the proteins interact, they put little magnets. So they make these cups, they put them in a big beaker, and they shake. In a minute, you have a sphere. Magic? No, just organization. Chance, it goes together, then it breaks apart, it makes a little one step more, it breaks again, and so on. They went further. They took a piece of plastic in a red color, and the one green, little bit different size. Then they built up a machine for shaking, because it takes more time. So shaking 20 minutes, you get at the end a red sphere and a green sphere. So not only did it assemble, but it sorted out which was the good one for each. So see, it looks magic, but this is just inbuilt in the way matter behaves. Now let me just show you examples, just a few, which are the ones we have been looking at, but I could have cited many others. What we will do here, we will use as bricks an organic molecule, a molecule. And we use as cementing entities which will bring them together, which will glue them together, a metal ion. So you have to choose right, you have to make the correct structure of your molecule, you have to choose the correct entity which binds them together. I don't want to tell you how you do that. And for instance, you can then make double helices, like we did here, or triple helices, and of course, you remember, this was the double helix of DNA. Now, let me say immediately that to scale, that's not to scale. These things are much smaller than that. But the principles of getting things together to form a double helix or a triple helix can be used also in other type of entities. And this is one of the things of chemistry, you can make things which nature never made, because there was no reason. Usually nature solved the problems when there was one to solve. But when the problem was not there, it didn't bother. It continued to evolve. But chemists and scientists in general can ask questions which nature never had to answer. Here's a nanocylinder. That's a bit more complicated. That's why I show it. Because it's formed of linear molecules, which are here artificially shown in red, planar molecules, which are the blue ones, and the connectors are copper, metals, these units here, these 12. So you mix these 19 components. You just mix, and you get the final structure. Many more complicated entities have been made by many laboratories around the world, and it just shows the power of understanding how things assemble and let them go. Since we are in an art institution, I'll show you some nice things. As you said, the computer allows you to give colors. Of course, molecules don't have these colors, but you can make colors. That's a nice one, which is sort. There are 5 molecules here: a yellow one, a red one, a green one, a blue one, and a white one. And there's something in the middle here. This is one which sort of self-organizes in a cyclic entity. That's another one, which is much more complicated than the other one. But you have the blue molecule, the green molecule, the red molecule, the yellow molecule, and in the hole in the middle, you have other things sitting. So you can also make yourself happy with looking at nice structures and giving them nice colors. Of course, the colors, as I said, are more for aesthetics than for anything else. This kind of process of self-organization following a given control, which you can build in by programming the system, is also interesting for nanoscience and nanotechnology, which is the theme of the meeting, which takes place right now in Bilbao, Imagine Nano. And this is a process which may in the future become more and more important. Why? Nowadays, we construct the companies making these fantastic devices all of us use, also your phones, your mobile phones in your pocket and all that. This is made by very fantastic technologies which make you such powerful things. This is true. That is enormous. I mean, it's incredible that you could achieve these kind of things. Storing, 30 megabits of information on a very small thing. This is something 20 years ago, these guys are crazy. But, okay, so this has to be made, which is fine. But maybe one could also think of letting the object make itself, which would, of course, be the ultimate way of fabrication. Just again to sort of convince you that indeed self-organization is at least one important way to go, the most complex computer in terms of complexity around is our brain. It's self-organized. You don't make it, it makes itself. Because the molecules know what to do, they know how to get together. So I think it's a line of development which can be very interesting, and probably the most powerful will be a combination of the fabrication approach and the self-assembly, self-organization approach. So just to give you an idea about what's going on now, this is now, it will be one, maybe two slides. What I showed up to now, briefly, is the fact that you can design chemical objects such that they can get together in the right conditions to generate a complex architecture. Controlling this from this organization by the information you put in the molecule, the way you program it. But a step further in this way, this path towards more complex forms of matter is to develop systems where it occurs with selection. Now, just in words, not in examples, it means that you can build up systems, and it's developing more and more now, this field in chemistry, where the system chooses by itself the bricks it needs to make itself. Of course, in that way, you need a diversity. You need a lot of different bricks from which to choose. And you need dynamics. You need these bricks to be weakly attached to each other. If they stick too strongly, you are blocked. If the thing is not the best meaning the most stable. So you use the possibility of acting on diversity in a dynamic fashion and to select the right brick to build up the object. This leads then to a chemistry which we call constitutionally dynamic. What does it mean? Complicated word, but it means just the following, is that the constitution of your chemical object, be it molecular or be it supramolecular, has the possibility to fall apart and reassociate. In other words, the constitution of the object is dynamic. It can break, reform, break, reform. If that is possible, it can also incorporate objects if you're not present at the beginning, and therefore do present adaptation. And so at least one of the things I am very interested in right now is to develop a chemistry which is adaptive, where the object you look at, if you change the conditions, changes itself and adapts to the conditions. I can't go further into that. So just the steps and then some conclusions in a more general way. The steps towards complex matter are the steps to life and to what we are. This self-organization process starts at the beginning with the bricks of matter. Of course, the elements are below this thing, of course. From the elements to make the molecules. This molecular chemistry. On molecular chemistry, you can build this chemistry which concerns the way molecules get together, what they do when they get together. I call it sometimes a molecular sociology, sort of what do societies or molecules do. This is design. You want to understand, you want to design, you want to see that you get the right object. In fact, when you don't get the right one, that's also interesting because you know you don't understand really exactly what is going on. So you try to think a bit more. Then you introduce these dynamics, and you get this chemistry which now is developing where the objects becomes breakable, that means we can dissociate and reassociate, and then it can perform selection, and an area is developing which one may call adaptive and evolutive chemistry. Now, this opens the door, of course, to adaptation, to many entities, to the diversity of the chemical world. But here, an artist, again, fits very well in this museum, but he was, in addition, an engineer and a scientist, Leonardo da Vinci. He wrote, for those who read Italian, look at the top, it's not so different from Spanish. And if you translate it into English, at the bottom, where nature finishes to produce its own species, we are part of it. Man begins. Yeah, that. Using natural things. What is that? We now know it's the periodic table of elements, the bricks which form matter, the universe. In harmony with its very nature, This is the laws of physics, the way things interact. And then comes something which for an artist is a very strong statement, and for a scientist also, to create an infinity of species. In other words, you open the world to all the possible combinations which can be made. Of course, some may be dull, others may be very exciting. So the essence of chemistry is, of course, to discover, to analyze what is around, but also to create expressions of matter which don't exist yet. So you must write the book of chemistry, not just read it. You must compose the score of chemistry, not just play it. So chemistry has creative power. And this fits again very well into a museum like here. And we will see another one. Here again, Rodin. The hand of the artist expresses out of the stone a sculpture which was not in it. So the stone is a stone, but out of the stone you can make a sculpture. It could be any other sculpture, but it is that sculpture, and it is that sculpture which is made by that person. And so chemistry is, in some ways, you can call it the art of matter. Let's consider science. Science is this inquiry of human beings into the universe, matter, what is around us. And the ancient mythology says that Prometheus stole the fire of knowledge from the gods. He conquered the fire and gave it to mankind. Consequence, we cannot give it back. He's here running, looking over his shoulder if the other guys are not running after him to catch him. But he gave us that thing. We cannot give it back. What you know, and you are stuck with it. I think it's very good, but we must know how to handle it. Now this fire, once he has given it, he shows it high. Our path leads us from the tree of knowledge to what we now can do to control our own destiny. Not let natural evolution do it, but do it ourselves. Now, of course, you may say that because we are the product of nature, taking care of ourselves is also a product of nature, which I think is true. That's a way to turn the opposition which says you shouldn't touch that. Touching what we are is a consequence of the fact that we are able to do it. And if we're able to do it, it's because we are what we are, and that's a consequence of the way the universe evolved. Lucas Cranach, a well-known German painter. At that time, people were painting many of what? Paradise, especially Eve. Here she is. What does she have in her hand? An apple. What did she do? She was picking the forbidden fruit of the tree of knowledge. This is the birth of science. Inquiry, knowledge, there is no forbidden fruit. And ladies, you are the first scientists. In the past, we were linked to the earlier entity, organism, in the way shown there. In the present, we can do this. Some of you must recognize this. Yeah? Michelangelo in the Sixtine Chapel. What about the future? Maybe. Now, this is a sort of a joke, so to say, but it's not so much of a joke. People may have lost their arm and have an artificial arm. That's what you do then. You can shake hands. We may have some titanium. This building is made of titanium. Some titanium in our hip. We may have new lenses in our eyes. We may have new teeth. Of course, these are small bits and pieces, but it's on the way of making us a combination of materials. And in the future, we will use what we know to do what we can about defects or amplifying even ourselves. There are biological ways of doing it, which you may be talking about in the discussion. So let me conclude by citing finally a mathematician. For the moment, my citing of mathematics was very primitive, was just arithmetics, 1, 2, 3, 4, 5. That's simple, huh? But David Hilbert is a very famous mathematician who is buried in Göttingen. And he has a tombstone there, and there is a sentence written on the tombstone which he wanted to have engraved. Here it is. What is it? Wir müssen wissen. We must know. Wir werden wissen. We will know. It's a strong statement. Huh? And one can discuss at length how far we can go, what does it mean we will know. But I think from the tree of knowledge to the light which is given us by science, probably I would say, certainly I shouldn't say certainly, because you never know. But there's a very good chance that we will know. And I hope that all of you in the room who are going along in this long trip to the future will contribute to it. I was very happy to be here in this fantastic building, one of the nicest in the world, and one of those which will remain for the 20th century. We had cathedrals in the Middle Ages. We have now buildings like this. And I hope you will enjoy it a long time and look forward to see you again. Thank you.
Thank you. Fantastic. The lights were not out because we need a minimum for the TV. So now, Professor Lehn will be happy to answer questions. El profesor Lehn estará encantado de contestar preguntas si las hubiese. Galderik balego, mesedez, aurrera there is translation, itzulpena badago, edozein hizkuntzetan.
Sí, buenas noches. Me gustaría...
Good night. Good evening. Could you talk about the inverse process? What does chemistry say about this when there's no more life left? Because I missed that you didn't speak about this issue.
Igual que pregunta se levanta porque están filmando.
I'm sorry, can you hear me? Can you ask him to repeat the question? Yes, I missed you speaking about inverse process in chemistry. You talked about matter to life, but what about the opposite idea?
We die. When you die, you become CO2, water, ammonia, some phosphate, some calcium, some salts, and that's what we'll all be. So life goes back to matter. Not very funny, but that's the way it is.
Hello, I hope you can hear the translation this time. Hello, can you hear me? I'm the interpreter speaking. Perfect. My question is very much linked to the first question. Because matter in itself, even if we talk about life matter, is not something that is really alive. And we've seen this with the virus and also with the death of people. Where do you find life? In matter, in the transformation of the matter, in the organization of this transformation. So where do we find life?
Is a consequence of more, it's an emerging property, as one says nowadays, that when matter becomes enough complex, at some stage it happens. Now, let me take this example. A virus is not living, it is a bunch of molecules. In order to live, which means to replicate and transfer its information, its genome, it uses some enzymes which are inside the cell. So in other words, we are very close to that threshold. We know that the virus itself is a bunch of molecules which lack the possibility to generate more of these molecules, which is due to some enzymes. It would be an interesting experiment, and I don't understand why biologists haven't tried it, to take an artificial bubble, what one calls a vesicle, to introduce into it a virus, to introduce in addition to that the enzymes, and see if it functions. In other words, a virus is just before being able to reproduce itself. Now, I also this question also comes from the fact that it is very hard to understand how things get living if we consider ourselves, because we are very complicated. But if you go down to the level of the virus just before being living, and the primitive cells like a bacterium or bacteriophage, this kind of less complex entities which exist in the living kingdom, then you can much more easily understand that it is possible. It's a question of the way in which molecules interact, the way in which they transform, and there is one property which is an answer also to the first question, that a living system is built on, let's say, the arrow of time, that things happen, the reactions happen, and life exists as long as the reactions work. When the reactions stop, you die. And then you decompose, because you go back to the fundamentals, if I may say. Yeah, I think no, we haven't been able to do it yet, but I think it's a property of matter. Life is a property of matter, but a complex state of matter, and there's now a very complicated, for those who are chemists in the room, all the self-organization I have described, and the one which chemists have mostly been doing, is self-organization in what is called an equilibrium condition. That means conditions where there is no movement forward, where everything is equilibrated, it's thermodynamically equilibrated. But now one tries more and more to couple this to chemical reactions so as to be able to give a direction to these assemblies. And that is then the beginning of having a system which can live. I have no better answers because at the moment it's not done. But I'm pretty sure that it's a question of doing it, of finding out how it will happen, of defining life also, because life is not human life. Of course, human life is life, but it's a very complicated one, as I said. And one has to look really down to the simplest type of organism to begin to think that we will be able to look at it and to see it. And luckily, the virus is just before being a living object.
Professor Lehn, thank you for your inspiring talk. My question, maybe continue your answer, previous question. You began from dark matter, dark energy. It is only 5% we know about universe. But my question, what do you think about dark matter of DNA?
Of what?
DNA.
DNA is not part of dark matter.
No, I mean it's like chemists told that only 5% of DNA active in living human beings.
No. 5% is the amount of visible matter in all the universe. And DNA is one part, is a molecule.
I just answer you. It's a question about that still it's not understandable the role of all sequences in DNA.
I think he means that in the DNA there is a lot of DNA that is redundant, that it does not work.
Chemicals call them dark matter of DNA.
You know, the DNA which is around, it's used, but this is why it is formed. You can make many others because you can make many sequences, and people are trying to make other sequences, even to increase and put a new letter in, to make it use this new DNA to have other functions, other proteins. So there are many, all the living organisms use DNA, so there are many different types of DNA, but not all of them. There are others possible. In fact, it's an interesting question to see if I modify one, and if I put it into a living organism, maybe it will be able to use it and to produce other proteins. In fact, genetically modified organisms, they sort of do that. It's now possible, for instance, for some of the biologics, that means the drugs, which are based on proteins, to use a genetically modified animal, like a goat, for instance, and make it the product you want is produced in the milk of the goat. The goat is doing another job, but this is still a DNA which comes from another living organism.
Okay, next. We allow for two more questions. Well, two more questions because it's already 9:30, and I'm sure there will be another 25. But please.
Have you ever thought about the energy and entropy of self-organization?
Yes, of course. Yeah, this is a very good question because, you say, of course, people say entropy, so it shouldn't get organized, but we exist. So first of all, you say we exist, therefore it works, number one. That's not an answer, it's a tautology, but it means that it works. Why does it work? Because what controls is not entropy, it's free energy. And free energy is the result of interaction, enthalpy and entropy. Which means that, for instance, the simple case of making a molecule of hydrogen, H2, can be represented as taking two atoms of hydrogen at infinite distance and bringing them together. This is a big loss of entropy. But the bond between the hydrogens is strong. So the delta H, the entropy, compensates for the loss of entropy. It's delta G which controls. But of course, the discussion is not so simple because the total entropy of the universe increases. But locally, you can have organization.
Okay, I think...
Who gives the energy for this entropy decrease?
Just the interaction between electrons, between the bond, the chemical bond. That's why it hangs together.
Is it enough?
Huh?
Is it enough?
Oh, yeah. Look, I can answer you. It's enough because it exists. But you can calculate it, and one knows exactly the energy of a hydrogen bond, of a HH bond.
Okay, I think I will ask the last question because you asked for
Really?
it. Yes, you wanted to give, you have strong views about genetic modified organisms, and you wanted to be asked about that. So I want to hear your strong views on that genetic modification. I'm sure the audience is looking.
But you know it already.
I know, but I'm sure the audience is looking forward to listening to your strong views.
No, the thing is, it's simple that we have learned how genetics can be used and how the genome of living organisms functions. And now we have the possibility to do very precise modifications which lead to the introduction of a given gene, that means a piece of DNA, into an organism, and which then makes it possible to give that organisms other properties. One example, and this I think, none of the objects you eat is wild. The tomatoes you eat have been modified genetically over centuries. A wild tomato is no good. A wild apple is no good. A wild potato is no good. A wild cow doesn't give enough milk. So, of course, it has been done by so-called natural ways. For a cow, you need a bull, and you need a cow, and you get a young cow, okay? And then you reproduce, and so this is fine. But you do something which in scientific terms is it's a bit a waste, so to say, because you use bunches of DNA, you mix the whole thing up, and you try to improve a given feature, like, for instance, making more milk, or for a tomato being nicer, better, and so on. So I have nothing against tomatoes which don't rot. And this can be done, has been made. Imagine, I go quite often to India. India has still regions very poor. In these regions, there are no fridges. There's no fridge. If you can make tomatoes which stay around for 2 more weeks, that's a big gain. You can eat them for much longer. There was also a case which, unfortunately, is, I think it's something very wrong has been done. A Swiss biotechnologist has developed a rice which is called the golden rice. Some of you may have heard about that, which is a rice containing the genes for vitamin A. And if children eat that, they get the vitamin E, which otherwise they wouldn't have. And there are many regions in the world where they need more vitamin A. So, and since you give me this occasion, I use it a little bit more. There is also a most fantastic thing. I would almost have become an embryologist. Because my first year at university, we had a professor of animal biology, which gave us a lecture on embryology, the experiments done by Spemann in the 1920s, for which he got the Nobel Prize. Spemann had done the following. When you look at the development of an egg that was sort of a lizard or batracien, what is that in English again? Like a frog or salamander eggs. And the eggs develops, 1 egg, 2 cells, 4 cells, 8 cells, 16, blah, blah, blah. And so it has different stages. And as it develops, one knows that one region of this egg will give the heart. Another region will give the neural spine, the spine. So he had the idea of saying, okay, I know that this part will give the heart. He took a little bit of these cells, put them on the other side of the egg, and he had 2 hearts. That's already very exciting. And then, quite recently, especially with Gurdon in the UK, and especially Yamanaka in Japan, succeeded in doing the following. In the development, our cells differentiate. Some give skin, the nervous system, all types of cells we are made of, muscle, heart, and so on. And these cells, for instance, skin cell, contains all the genetic information of any other cell, except that in the course of development, some of these genetic expressions have been repressed so that the cell has evolved. One has given skin, the other one has given heart, and so on. And Yamanaka was able to, as one calls it, de-differentiate. Differentiation is from the initial cells, which are totipotent first, or pluripotent, that means growing any tissue. He has been able to find, it's rather simple at the beginning, 4 factors, 4 proteins. And with these 4, it was a real hard work because I had to try lots of things. He was able to take cells which were differentiated and put them back to the state of stem cells, to the point that he took a cell from the skin of a mouse and he made a mouse out of it. You see what that means? That means that you begin to understand how one can take any cell of our body and make our organs. That is why I also think when I showed that picture with the robot shaking hands, this is one possibility, but it's possible that a much more exciting one is to use developmental biology where rather than having a new heart coming from a donor, you make your heart. If we understand someday how a cell can be pushed into not making a full organism, but just a tissue. Skin, you burn yourself. And people who have very much burned, usually you take another skin from somewhere on the legs or something, you put it on, but you will be able to make the skin. You will be able to make the heart. It's not science, it sounds like science fiction, but there's a good chance that it will be possible. So I think, we are the result of evolution. We have been put together in a way which has built on each other. We are a Lego box, a complicated Lego box, but we understand how it functions. And for instance, there's Walter Gehring in Basel. He did things which some people jump up when you hear that. He made flies which had legs in the place of the antennas. Because the insects are much simpler. They have regions of the body, and you can sort of use this to play with it. Or he also made flies which had eyes everywhere. The fly had eyes on the legs. And it seems that even they could see with that. This would be fantastic. You could have some eyes on the back of your... I would like to have 4 arms. You can play piano much better.
Okay, we must know, we will know. Thank you very much, Professor Lehn, and thank you, all of you.